Welcome to a small introduction of the lab. So the first thing you do when you enter a lab is you wash your hands. Wash them a little bit as if it was still Corona times, as we want to make sure our hands are as clean and sterile as possible before working with our cells. We want to avoid, of course, contamination with any possible bacteria or fungus. Make sure to rinse off all the soap. And then to properly dry your hands. Once you have washed your hands, you can put your lab coat on. And it's this lab coats with a green neck. And the green neck indicates that there are lab coats only for the ML1 or the ML2. So please avoid taking these lab coats outside of the lab. So we'll start a tour with the CO2 incubators. These incubators is where you will be putting your cells and where they will be left overnight to incubate and to grow. And these incubators are kept at 37 degrees with a CO2 level of 5%. And this temperature and CO2 level is optimal for the cells that you will be working with. So then we have our laminar flow cabinets. They're all around the lab. And in the next video I will explain how to operate the laminar flow cabinet and what the different components are inside and outside of the laminar flow cabinet, and that you have to keep in mind when you work with them. In the middle of the lab we have different machines that can be used, different equipment. We have a centrifuge which will not be needed, and we also have a Bio-Rad automated cell counter used for counting the amount of cells in a sample. This equipment will be explained in a later video. In a small corner, dark corner, we have some microscopes. They will also be explained how they work and what the principles are in the next video. But the location of the microscopes might vary depending on where you're working, so which cell culture you're working in. Then we also have a heat plate and a water bath, which will be used to keep small samples or small media and also bigger flasks warm throughout the day. We also have some racks here, which again, the location will vary depending where you're working. And yeah, you can find materials that you might need, like tubes, for example, some 15 milliliter tubes or 50 milliliter tubes. Also, you might need it at certain points, PBS. If you know where it's located and you see that assistants are pretty busy, you can just grab it yourself. Otherwise, just wait for the assistants and they'll happily grab whatever you need for you.